using in vivo MRI techniques to reveal circuits that are differentially sensitive to traumatic stress in male and female rats. So there's less about references and more about keynotes. Okay, so PTSD is a debilitating disorder. It's something you experience after traumatic stress. Not everyone experiences PTSD-like symptoms, but to get a diagnosis, they have to last for over a month. So it's about like 6 to 7% of the population, 30% in people exposed to war. It goes up the more traumatic the event is. Surprisingly, or not surprisingly, it's more prevalent in women, but the reason for this is unknown. Why do women experience PTSD more when, statistically speaking, they're less likely to get exposed to trauma? So, that's a big push in the PTSD field right now, traumatic stress field. Why are females more susceptible to traumatic stress? So there are a number of ways you can tackle this, but one way to do it is use an animal model, because you have a lot of control over the variables. You can manipulate circuits or hormonal systems in ways that you could not in a clinical study. And there's enough evolutionary conservation in a vertebrate system, or actually even in a Drosophila, to inform clinical practice. Identify circuits, receptors, molecular pathways, mechanisms that could make women more susceptible to developing PTSD after trauma than men. So that was the point of this study. And what is the impact of traumatic stress on male and female circuits in the brain? We wanted to start. I don't want to be too how to say, over dramatic, but starting from ground zero. You'll see what I mean. So the blueprint was, a problem that comes up is, where do you start, right? Like, what circuit do you look at? How do you know a circuit is conserved between male and female? How do you know something that's not necessarily important for fear or anxiety is driving the sex difference? You just don't know where to look. So our animal model is one I helped develop: single prolonged stress. It's serial exposure to restrained force women ether. It's a good model. I could put lots of references here that shows it's a good model, but in the interest of time, what we wanted to do was use this model and do a hypothesis testing and a data-driven approach. Okay, so the first one, we use in vivo MRI, and it's great because you could use it in humans as well. And so anything that we identify is readily translatable into clinical literature, and so that's why we were excited about it. So the default mode network is this collection of brain regions that seems to be always on. Like my computer I'm presenting on, I have to turn it on. For humans, as long as you're alive and you're not in a coma, we seem to have these midline brain regions that are always on. If you're doing, a magnet, doing nothing, they turn on. You're sleeping, they're on. You're under general anesthesia, they're on. When you engage in a task, they turn off. So that's why they're referred to as default mode network. They're disrupted. That network is disrupted consistently in PTSD. Now, rodents have a DMN too. It's not really clear why the DMN is disrupted or what it means, but we wanted to examine the effect of SPS on that. And then emotional networks are consistently disrupted in PTSD, so we wanted to examine that as well. And the approach that we took was this before and after. This is something you typically don't have in a clinical PTSD study, where you have rats, you analyze them for resting state functional connectivity, do your stress protocol, and then you examine how stress changed functional connectivity, as well as in control animals the passage of time. Everything that I'm about to say, we think the power of this approach is this before and after. You could document, there's no behavior, there's nothing going on. It's purely data-driven. How does stress impact functional connectivity across the entire brain? And then if there was an effect, was there a change in volume? Right. So we found that SPS disrupts default mode network connectivity. Okay. We check to make sure our brain is correctly registered in the scan. This is functional connectivity. It's a correlation of bold signals from one brain region to every other brain region. So I think there's 177 nodes here. This is the rodent DMN. here, we put every connectivity into a vector and calculated the length of that vector, which is just the strength of connectivity within this network. And it doesn't really change for control to kind of hovers around 0, but it decreases for SPS animals. So, it was more of a verification. SPS decreases connectivity within a DMN. It would appear that this is not a predisposition to developing PTSD. People with low DMN connectivity, more susceptible to trauma. This is something caused by trauma. So then we had a series of filters in the emotional network where we made it more stringent. We subtracted post from pre, and then this is everything. And then we said, okay, we only consider things that are greater than 0.1, 0.15 until we get to 0. And then we look at this penultimate network, right, in males and females. This is what it looks like. And then we subjected these brain regions, these connectivities, to a factor design. And this is what we came up with. So purely data-driven. We're not assuming something happened somewhere. We're just letting the data and these algorithms lead us to where it leads us. And this is where it led us. In male rats, you have an increase in thalamic ventral hippocampal connectivity and controls that are absent in the stressed animals. And this is only for males, not females. In female rats, in stressed animals, you have an increase in thalamic ventral hippocampal connectivity that you don't see in males at all, and you don't see in controlled females. So interesting, right? There's a sex difference circuit that we observed. Now, all the brain regions that were affected, I'm not showing all of the data. We then plugged them into a simple factor design and looked at changes in volume, and then corrected for false discovery rates. The only thing to survive was the dorsal dentate gyrus. So we took an anatomical scan as well, and we have software that allows us to measure the volume. So if you look at control rats, there was an increase in volume from that pre-scan to the post-scan. So the dentate gyrus got a little bigger, and that was absent in SPS rats. So you can see the effect is bigger in females, but it's a main effect. It's not really like an interaction. So, what did we know? SPS, traumatic stress, disrupts DMN connectivity. It's not a predisposing factor, something caused by trauma. Thalamoventral hippocampal circuits, and this is completely new, right? I could not find much literature on what their connections, like physical connections between the medial dorsal thalamus and paraventricular nucleus, the ventral hippocampus. No one knows what those neurons do, what this region is important for, but they could represent circuits through which sex differences manifest with regards to traumatic stress. And then the hippocampus is this big area in PTSD. What we've observed is something a little different, is that in adults, the dentate gyrus might still be growing. And what traumatic stress, in this case SPS, may do is prevent expansion of his dorsal dentate gyrus in adults. Okay, so that's the take homes, that's the conclusions, that's what we could say. So I can't do all of this by myself. Clearly, these are the co-authors that helped. DE-INBRE was super helpful in making this possible. COBRE grants as well, these idea funds. Frontiers Award from the University of Delaware, that was helpful too. And then the state of Delaware was super, super helpful. I really appreciate it in these trying times of scientific funding. And I believe Center for Biological and Brain Imaging, to try to learn this new technique on the fly. We started during COVID. It was instrumental to have a team that can help us. And there's a good team set up at the CBBI.